So first of all, just to set the foundation here, of course, key objectives of drug discovery really are summed up by the idea of multi-parameter optimization. What we're trying to do is identify chemistries or compounds with an optimal balance of properties that are required in a safe and efficacious drug. So not just potency against our target or targets, but appropriate physicochemical, ADME, and safety properties. They all need to come together in the same compound in order to have a high quality lead and ultimately candidate drug. And of course, we often start a project somewhere out here, typically with a reasonably potent hit or lead compound. And our goal in the course of a project is to move as quickly as possible from that starting point to this sort of sweet spot in that chemistry space that's going to deliver us that high quality, successful candidate drug. On the other hand, we also want to identify situations when that balance is not possible. Essentially, when the space that gives us potency against our target does not overlap with all of the other properties we require. And of course, this is very commonly described as, fail fast and fail cheap. Don't waste time and effort making and testing compounds that won't succeed downstream. But that's a very negative message. I've never met a project team that wants to fail. So we want to add something to that, which is we only want to fail when we are confident that that's the right decision to make. Essentially, when all the data we generate can convince us beyond a reasonable doubt that there is no good drug in the chemistry that we're exploring, then it's appropriate to move on to the next round of chemistry, maybe a different target or even a different project. The reason we highlight the word confident is it's something we'll come back to a little bit later, because we need to recognize that there's significant uncertainty in all of the data we deal with in drug discovery, experimental variability, and statistical uncertainty in predictions. And the last thing we want to do is to miss the opportunity to find a drug by incorrectly discarding a compound due to an incorrect measurement or inaccurate prediction. So with that in mind, what are we looking for in a method for multi-parameter optimization in drug discovery? Well, first of all, it needs to be interpretable. No one likes black boxes. We want to understand why a compound has been given a certain priority. And more importantly, if there are issues identified with that compound, what do we need to modify and change to improve that compound's chance of success? The method needs to be flexible as well. Every project is different. There's no universal set of property criteria for every drug discovery objective. So as a project team, we need to be able to define the criteria depending on the therapeutic objectives of our project. Is it CNS or peripheral? Is it an orally administered compound or IV or inhaled? Is it a chronic or an acute therapeutic that we're looking for? And many other requirements. We also need to be able to weight the different importance of different endpoints, different things we measure or predict to the success of our project. Some things are absolutely critical to success. We must have a potent compound or there's no point going forward. But other properties we might be willing to compromise or trade off against other more critical factors. And then, as I mentioned, we need to take that uncertainty into account, the experimental error or variability to avoid those missed opportunities. So with that in mind, there are many different methods that have been applied for MPO in drug discovery over time, and I've sort of summarized some of them here in a list. I've highlighted a few that we'll focus on in more detail, so looking at filtering, desirability function, probabilistic scoring. But just before I move on, I thought it was just helpful to summarize the other three in this list. And the first of those are sort of simple rules of thumb that we often follow in drug discovery. The most famous, of course, is Lipinski's rule of 5 for orally available compounds. So 4 simple criteria based on simple characteristics of a compound that we can calculate. But there are many, many others. There's the 375 rule by Hughes et al. There's the rule of 3, and there's an enormous list of different criteria that people have proposed. These have some big advantages. They're very simple. They're very easy to apply. They're very easy to interpret. If the log P is too high, you can aim to reduce the log P of the compound you're exploring. But and there are some very big buts here, is first of all, they're tailored to specific objectives. Some people have treated Lipinski's rule of five as a definition of drug-like compounds. But of course, these were proposed only for orally available compounds. So we need to make sure we're applying it in the right context. And there's also a risk of too rigid application. These are guidelines. They're not hard and fast rules. If your compound has a log P of 5.1, that's not really different from 4.9. So we need to be careful not to inappropriately discard compounds. And even Lipinski himself describes these as sort of rules of thumb or guidelines. They're not hard and fast filters we should apply to our compounds. Another common approach is to sort of combine multiple parameters into a single metric that we can optimize. So very common here are things like ligand efficiency and ligand lipophilicity efficiency, and there are many other ligand efficiency indices that have been proposed. Also, very popular now is human dose estimation, combining things like the clearance, the potency, and so on into a single estimate of the dose would required in a human patient. Now, again, these have the advantages of typically being quite easy to interpret, at least usually, because there is sort of one parameter to monitor, and we can see how that is calculated from the underlying properties. But again, that has some disadvantages. We need to be careful about not applying them overly rigidly. They are rules of thumb. There are some questions about the validity of some ligand efficiency indices, and there's a huge literature debating that. They are, again, often tailored to specific objectives, and that may not be transparent. And also, they don't include many different important factors. So the ligand lipophilicity efficiency is helpful because it just combines potency and log P, for example, but those don't capture all of the different factors that we need to optimize in a safe and efficacious drug. Even human dose estimation, although it brings more factors together, still omits many important things that we do need before we can nominate a clinical candidate. Another method is called Pareto optimization. So that's based on the principle that there's not just one optimum, but many that are equally optimal in a combination of different properties. So here you can see I've plotted just 2 dimensions where I've got the potency and microsomal stability, and you can see the position in this space of different compounds. And the Pareto compounds are those that occupy this so-called Pareto front, i.e. they're what's called non-dominated, or there is no compound that is better in both dimensions than any of these compounds. On the other hand, you can look at this compound here, and you can see that the orange compound just above and to the right is better on both of these axes, so this is not a Pareto optimal compound. Now, this is actually a really nice approach if the appropriate balance you're looking for is unknown sort of in advance. We might want to explore all of these different trade-offs between potency and microsomal stability to find those that actually deliver the best outcome downstream. It's very flexible. You can define any combination of parameters that you want, and it's easy to interpret in a plot like this, for example. But it does have some disadvantages. It can be overwhelmed by large numbers of parameters. For example, if you get more than 5, there are so many compounds on the Pareto front that actually you're just left with a slightly smaller problem, but still having to choose between many, many different options, not knowing which ones are the best. And it neglects uncertainty. We've got to remember that every single one of those data points has uncertainty around it. An error bar is shown here. And so can we really say that, these compounds are actually, none of these are really within statistical significance better than each other? Which one in practice should we pick? Now, these and many others are discussed in much more detail in a review article. So if you're interested in that, we'll also provide the reference at the end. But let's look at a few of these as an example and show actually how they can get quite different results here. So in this case, I've got just a very small data set, just 9 compounds with some properties here is its potency, it's a pKi, solubility, log P, hERG inhibition, and blood-brain barrier penetration. And we have a combination of experimental and calculated values for each of them. The calculated values are the ones highlighted in green. And if we have a set of criteria that we could apply as filters, so for example, we want a pKi of greater than 9, we want a log S of greater than 2, better than 100 micromolar, log P less than 3.5, and so on. We can apply these filters. And if I do that, what we can do is I've just removed here the cells that don't meet those criteria, leaving the ones that do. And you can see, for example, well, none of these compounds meet all of the criteria that we're looking for. So now we're left with a bit of a trade-off. You can see here that the compound in row 9 actually meets 4 of the criteria. It's the only one that does, so we might give that a rank of 1. You've got here, this one meets 3 criteria. You've got several that meet 2 criteria. Which ones are best? And how important is each individual criteria relative to each other? How do we trade those off, given that none of them meet all of those requirements? So it's a really much more complicated problem, and we can rank order them, but is that the right ordering? Now, instead of these sort of hard filters, just greater than or less than, we can represent the outcomes for each property in terms of how desirable that outcome is, commonly described as desirability functions. And these date back a very long way, and not within drug discovery, but they date back all the way to the 1960s. And so if we're thinking about a simple filter, so in this case, we've got some property and we've got a desirability on the y-axis, we could look at a simple filter greater than 5, and that would be represented by this step function here. So greater than 5, it passes, it gets a score of 1. Less than 5, it fails, it gets a score of 0. So that's, again, representing a filter as a desirability function. But we can now do something a bit more subtle. Instead of a hard filter of good and bad, we can actually set a lower value, but not 0, for compounds that fail, in quotes, to meet that criteria. That can represent that it's desirable to be greater than 5, but not catastrophic to be worse. And we can think about the importance of a property in terms of that difference between the ideal score of 1 and the lowest possible desirability here. A very important property will have a very low desirability if we fail. So this difference will be large. But a property that is nice to have but not critical would still be quite desirable even if it failed, so it would be up here and that difference would be small. So we can represent those trade-offs that we're willing to make between different properties. But now we don't need just simple step functions. We can be more subtle now. So here we can have a trend. So ideally, the value we're looking for is greater than 8. That sort of passes and gets the perfect desirability of 1. The worst outcome is less than 2. That gets a desirability of 0. And we've got a trend. It gets better and better and better until we reach that ideal outcome. Or we could represent a range maybe with soft boundaries. We can get as sophisticated as we like with this. So here the ideal range is between 4 and 6, but we've got these sort of soft edges to that. And though you can see here, it's worse to be above 8 than it is to be below 3. So you can define all sorts of different shapes of these desirability functions. So really, the reason for using this is to avoid hard cutoffs that draw artificially hard distinction between similar compounds. You can apply these soft boundaries to those ideal ranges to represent the fact that if you're just outside, it may not be catastrophic for the outcome of your project. So, one way we can look at this is, well, what impact does a particular value have for a property have on the ultimate outcome of your project? And so here I'm just showing an example based on CACO2 permeation. So this is actual experimental data. It was published quite some time ago. But here we have again, the clinical human intestinal absorption represented as a percentage against the CACO2 apparent permeability here on the y-axis. What you can see here is that if we have high permeability, so we're high on this scale, we're very likely to have high human intestinal absorption. But if we have low CaCO2 permeability, actually, that doesn't tell us much about whether that compound is likely to be absorbed or not. There's quite a range of values here for the observed clinical human intestinal absorption. So we might turn this into a question of, well, if we're ultimately looking for human intestinal absorption greater than 50%, what is the chance of that compound achieving that outcome, given a measured value for the CaCO2 permeability? And so we can turn this into this sort of graph here. We've got the CaCO2 permeability and the proportion of observed compounds meeting that criterion. So again, high permeability, we're pretty much guaranteed. That's this group of compounds here in the top right of the scatter plot. But if we have a low permeability, you can still see about 30% of those compounds meet that criterion. So we wouldn't throw away a compound simply because it had low permeability. It still has a reasonable chance of success. And we can represent that as a desirability function representing that likelihood of success, shown here by the blue line. And we could use that in our prioritization of compounds based on CaCO2 permeability. So if we go back to this very simple example data set, instead of these hard cutoffs, we might actually define desirability functions for each of these different properties. So here's a trend. Here you can see a hard cutoff. Here's a range and so on. And if we apply those desirability functions to each of those values, we can actually calculate a rank based on that. In this case, it's basically simply taking the multiplying together the different desirabilities of each of those properties and then ranking it based on the highest product of all of those. And here you can see quite a different outcome. If you recall, looking at filters, what looked to be our best compound here was compound number 9, but with desirability, it's actually completely different. It's compound number 1, and compound number 9 actually ranks quite low on that list. Excuse me. So instead of simply filtering, using desirability functions, we can get really quite a different result. But this is actually neglecting another very important point, which is the fact that we don't know each of those property values exactly. If we think simply about the experimental measurements that we make, then we need to think about the variability in our assays and experiments. So for example, if we have just a single measurement, we might look at the assay variability, and a pKi or PIC50 might have a variability between 0.3 or 0.7 log units. That is a factor of 2 to 5 in the actual Ki or IC50. And of course, later on in a project, we might have multiple replicates measure the same compound in the same experiment multiple times. And of course, we never get exactly the same answer. We get a spread of different results here. And we would typically represent that as a mean value and a standard error in that mean. There is some probability distribution for that mean value that we can calculate. So we can put error bars on that. And of course, if we're looking at predictions, then there is significant statistical uncertainty in those predictions. If you look at a typical log P model, the standard error in prediction is between 0.4 and 0.5 log units. And if we're trying to predict, potency, pKi, for example, then we might have up to a log unit of uncertainty. That's a factor of 10 in the Ki. Here on the right-hand side, I'm showing you a typical sort of solubility model. This is observed versus predicted. And of course, there's scatter around that. It's about a 0.8 log unit root mean square error, or average error in that prediction. What does that mean? It means that we can't really tell between 1 and 5 micromolar values of solubility. We might have a reasonable chance of telling between 1 and 10 micromolar. So we need to use that with an appropriate degree of resolution. So when we got this uncertainty, we need to think about the impact it has on the decisions that we're making. So if I think about some property, we'll call it Y, and we might have some threshold. On the right-hand side is desirable. On the left-hand side is undesirable. If we were to look at 3 measured values for 3 different compounds, A, B, and C here, If we were treating this as a hard filter, then of course what you'd say is, well, compound C meets our criteria, that one passes. Compound A and B fail, so we would throw those away. We would reject them. But of course, that's neglecting some very important information. If we look at compound A, for example, of course, we don't know that value precisely. As I just said, we actually have some probability distribution. Now, here you can see the probability of this value exceeding our requirement is negligible, so that's fine. But for compound C here, you can see, although it looks to be the best, actually there is a significant probability, the sort of region to the left of that cutoff, that it is not going to be good enough for our requirement, shown by that shaded region. And if we look at compound B, although it looks to be the wrong side of that threshold, again, there's this shaded region, this probability that it is actually good enough for our project outcome. And so when we're looking at these three compounds, A, B, and C, what we really should be saying is, yes, you can reject A. That is very unlikely to achieve our requirement. But projects B and compounds B and C, they're in a gray region. We can't really choose between them based on the data we have so far. We would need higher precision results, maybe multiple experimental replicates to be able to make that decision in practice with confidence. And if we look at another very simple example here, I've got our same cutoff. We might have these two compounds, D and E. Both of them are the wrong side of that threshold. But what if they were the two best compounds for our project and we had to pick one of them to go forward? Which one would you choose? Well, you might look at compound E and go, well, that's closer to the threshold. That's the one I'm going to choose. But again, we're only looking at part of the information. If we look at compound E in more detail, actually, we have high confidence in that, a small error bar. That probability distribution is very tight. So the probability of exceeding that threshold is very, very small. But maybe compound D has greater uncertainty. Maybe we haven't got multiple replicates for that. It's got a bigger error bar around it. So in fact, that probability of it exceeding the threshold is actually larger than compound E, shown again by that shaded region. And so in that scenario, what I would suggest is you'd actually prefer to progress compound D because it has the higher probability of achieving the outcome you're looking for. Of course, the first thing you would do is go and measure that in more detail so the error bars become smaller, and then you'll be able to make a confident decision. But if I had to invest my time and effort, that's where I would put it. So we can bring all of this together in a multi-parameter optimization approach. We call it probabilistic scoring, so that you can define not the profile or properties you're looking for. You can define not only hard cutoffs, but also desirability functions that define the sort of trade-offs that we're willing to make, and not simply hard cutoffs. And we can combine that with the uncertainty in the information that we have, the property data that we have, to calculate an overall score that represents the likelihood of success of our compounds. So we can take that property data, it can be experimental or predicted. We can assess it against our criteria for success, defined as desirability functions, which capture the relative importance and trade-offs of those properties, and the uncertainties in the data, whether experimental or statistical. Calculates not only the likelihood of success, the score itself, but also a confidence in that overall score. Now, given the uncertainty in the data, how confidently can we resolve and choose between compounds? And one way to look at that is shown here in the bottom right. This is something we call a snake plot. What I've got here are the compounds ordered from the best on the left to the worst on the right. On the y-axis, I have the score itself. And here you can see the best scoring compounds in this data set have about an 80% chance of success. Yeah, we'd be really happy with that. But what you can also see is an error bar around each of these scores. That's showing us the confidence in that overall score once we combine all of that uncertainty in the underlying data. So what we can actually see, for example, is that the data we have do not separate those top 5 compounds. The error bars overlap almost completely. If I were to tell you compound 1 is the best, go for it, you'd go, oh, wait a minute, you haven't got the evidence for that. I'd need more data, or better data, so the error bars become smaller, or another criterion with which to choose between those compounds. On the other hand, roughly the bottom 50%, you can see the error bars don't overlap. We can say it's very unlikely those are going to be as good as our best compounds. We can reject those with confidence and focus our time and effort exploring the chemistry at this end of the scale further, generating some additional data before choosing the way forward for our project. So if we actually look at this toy data set we were looking at again, so again, now we have these properties with our desirability functions. We have some estimates for the uncertainty in those values as well. We can combine that with this probabilistic scoring approach, and that enables us to prioritize our compounds in terms of the probability of achieving the ideal outcome that we've defined for each of these different properties. We get this overall score. And also you can see these histograms that tell us the impact of those different properties on that overall score. So a high bar is good, very, very likely to achieve that outcome. A low bar is one that is very unlikely to achieve a very important outcome. So a critical issue for that project. And again, we get quite a different order for the compounds in this very simple toy data set. In fact, now rank number 1 is actually row 3, which has the highest probability of simultaneously achieving the ideal outcome for all of those properties. And where it has a potential shortcoming against a property, it's not a critical property or it's not a high probability of failure. Okay, so that's all well and good. That's a lot of theory there, and a really simple toy example to show you the impact of these different approaches on how we would choose our compounds for progression. So let's actually delve into a real case study that shows this in action. So to give you a little bit of context for this project, this example is based entirely on experimental in vitro data. So no calculated values or predictions. These are actual experimental measurements. So 150 compounds had been measured in a combination of in vitro potency, selectivity, solubility, and microsomal stability. So a really rich data set for this project. But previously, compounds had been selected for study in vivo studies based primarily, almost entirely, on the selectivity and potency, ignoring potential issues around solubility and metabolic stability. And what had been found in practice is poor bioavailability in the rat models in which they were studying the compounds. And so the goal here was to select compounds with a balanced set of properties for progression to in vivo studies. And here's a sort of a summary of those criteria for this project. They were looking for selectivity, so better than 8-fold selectivity, potency of less than 1 micromolar, solubility greater than 10 micromolar, and good stability or low turnover in human and rat liver microsomes. And these are sort of ordered from the most important at the top to the least important at the bottom. So actually the critical challenge for this project was achieving selectivity against an off-target in this case. And so that was actually given the #1 priority. Now, let's just have a look at the sort of historic approach. So basically filtering based on potency and selectivity. So looking for selectivity greater than 8-fold and potency less than 1 micromolar. And so these have just very simply ranked against the measured values for both of those properties, ignoring potential errors in that data and uncertainties. And on the right-hand side, we're just showing the top 15 compounds ranked by this approach. And I'm highlighting one compound here, this one 572, as an example that we're going to follow during this case study. So this achieved both the both the selectivity requirement, so it was 11-fold selective, and the potency requirement, so about 200 nanomolar potency. But it wasn't the most potent, and it wasn't the most selective compound, so it hadn't been chosen for progression. It wasn't in these sort of top-ranked compounds. So let's just take the uncertainty of that data into account. So we use some estimates of the uncertainty and the selectivity and the potency, and simply using the same cutoffs, the same criteria, we just applied that uncertainty and look at the probability of achieving that outcome. What you see, first of all, is this compound we're following, 572, has jumped from number 14 up into number 7, because in terms of the probability of achieving both of those outcomes, it is actually better than others that were measured to the specific values were measured. But I'm just going to highlight here compound 561. Previously, this was number 28 on the list, so right off the bottom of the screen. It's jumped up here into contention. It's not in the top 10, but it's number 12. And we can see why. It's actually one of the most potent compounds this project had measured, so just over 100 nanomolar, so really very potent. It had a selectivity of 7-fold, so it didn't quite meet the requirement of 8-fold selectivity, but selectivity is a very uncertain measure. It's actually the ratio between two potencies, and so that has a really big error bar. So in terms of the probability of achieving both of those, it is worth considering, particularly given the potency of that compound. So it's in contention, but not right at the top. But now we can look at the effect of taking all the in vitro data into account. So not just potency and selectivity, but solubility and microsomal stability in human and rat liver microsomes. And now what you see, if we look at this third approach, first of all, this compound 572 jumps right up to the top, number 1 on the list. And if we look at that, you can see why. Again, we saw it's very selective. It's very potent. It's very good solubility. It's very stable in human liver microsomes. The only question about it is the stability in rat liver microsomes. That is higher than we would like, but remember, that is the lowest importance criteria for our project. And so in terms of the overall trade-off and balance, that looks to be the best compound. And I've also just highlighted another compound here, 518. This came second. So previously, it was just completely off the bottom. It was number 52, the last, looking purely at potency and selectivity. But it's jumped all the way up to number 2, because although it doesn't meet the selectivity criteria and doesn't quite meet the potency criteria, given the uncertainties in those, there is a probability that it will be good enough. But in terms of the other properties, it was one of the most soluble compounds measured. It was rock solid in human liver microsomes and stable in rat liver microsomes. So in terms of that overall balance of properties, it is certainly worth considering and not rejecting outright. So again, using real data here, you can see quite a difference in the way that we would choose the compounds in the context of this project. And in terms of the outcome, the study identified 4 compounds that had been completely overlooked by this more sort of traditional method based on selectivity and potency cutoff. And in particular, that compound that was number 2 on the list, 518, it was taken forward to be tested in vivo, and it was found to have a superior pharmacokinetic profile. It overcame those issues with bioavailability that the project was having. Even more exciting for the project was that it was the only representative of a novel chemotype. They tried it in a bit of a hunch. They'd synthesized and tested it. It wasn't found to be sufficiently potent or selective enough, so it was just rejected and never explored further. But now, knowing that this chemotype gives them reasonable potency, reasonable selectivity, but achieves the good ADME and pharmacokinetics, it gave the project a new route forward to optimize the potency and selectivity, which is something they knew how to do very well, knowing that they were in this sort of good ADME, good PK area of chemistry. And it was overlooked completely without applying this multi-parameter optimization approach in an effective way. Now, just before we wrap up the slides, there is a very common question as well. What if I don't know what the right criteria are to choose compounds for my project objectives? Now, sometimes, of course, we know based on experience, we can estimate those criteria that we're looking for and have that discussion as a project team and decide the profile for an ideal compound. But sometimes the data is too complicated to be able to know that a priori. And so there are approaches to finding these sort of tailored profiles. So you can use existing data to actually find scoring profiles that identify compounds with an improved chance of success. And success could be against any drug discovery objective. It could be as grand as success in the clinic for a particular therapeutic indication. It could be achieving appropriate PK, avoiding toxicity, anything that you'd like to define as an objective for which you have compounds that were good and bad. Of course, once you've found that scoring profile, you can apply it prospectively to new compounds to find those most likely also to achieve that objective. And any data can be used, whether it's experimental or predicted. And to do this, it explores multi-parametric data. It considers the combination of properties, not individually. And this is important because it avoids overcounting of correlated factors. And it generates rules that are both interpretable and modifiable. Again, we want to avoid black boxes. We don't want to, have some method where, just says good or bad, well, why? And equally, given the limitations of the data we often have, we also want to be able to tweak or modify those based on our experience and get that synergy between computers and experts. I've sort of illustrated this here in two dimensions because I can draw two-dimensional space, but we've got two properties here, and we're looking really for regions of property space that have predominantly good compounds and very few bad compounds in that space. And you can find potentially multiple regions, different trade-offs of properties, and each of these corresponds to a scoring profile in the way that I just illustrated. Now, the way that we can do this is a method called rule induction. It's actually called the patient rule induction method. It's a way of actually exploring these multi-parameter spaces to find these regions, these boxes in property space with a higher chance of success. If you want to know more details, there's a publication on the previous slide. Again, we've got some references at the end to see in detail how the algorithm works. But essentially what the algorithm tries to do is to maximize the mean value in this box. So maximize the number of good compounds relative to bad, or a continuous parameter, a value that we're looking to increase or decrease. We also look at the proportion of the data set covered. There's no point having a box that has just one good compound in it. That's a perfect criteria, of course, for that one compound, but doesn't generalize to finding new compounds. And so this is a conventional sort of specificity versus sensitivity trade-off. And you don't have to fit just hard boundaries to these boxes. Again, these sort of step functions, you can actually automatically fit these soft edges to the ideal regions that the method finds as well. And this is in what we call our MPO Explorer module within our StarDrop software. Okay, so again, that's a little bit more theory. So let's take a few minutes before we get on to questions, just to actually see what this actually looks like in practice. And what I'm going to do is use our StarDrop software to show you an example using a data set, a slightly more complex data set than the ones you've seen so far in the slides. So this is StarDrop. I have a typical sort of data set loaded in here. What this has is about just under 300 compounds. We have the structure and the identifier. We have an experimental potency. So this is PKI value here against the target, which is 5-HT1A in this case. And we have a range of different calculated properties for physicochemical and ADME properties in which we are interested. Now, if we were looking only at potency, we can immediately see here from this box plot on the right-hand side that the most potent series, at least on average, is this one here. If I select it, you'll see these are all indenols. And so if we were just purely looking at potency, that's the series we would focus on because, yeah, it's the most potent series in this in this library of compounds. But of course, we've been talking about not just using one criterion or one property, but looking at multiple criteria. Now, of course, we can take this data and start to plot some nice pictures. I could plot the potency against the solubility, and I could sort of add in there the blood-brain barrier penetration and generate this sort of nice spinning sort of 3D scatter plot. And we could add more dimensions to this. I could color it by ERG inhibition. I could change the size by another property. I don't know, 2C9 inhibition. I don't know. You can create these crazy plots with different symbols and sizes and shapes and so on. But quite frankly, I have no idea what you do with a plot like that. How do I actually look at that and say, oh, yeah, they're the best compounds for my project? It's almost impossible to decipher. I've got, what, 5 dimensions of data plotted there at the moment. So let's forget about that because, of course, what we've been talking about is a way to solve that problem, which is to use this sort of multi-parameter optimization approach. So let me go into this sort of scoring area where we can construct a scoring profile that represents what we are looking for in a compound for this project. Now, I could start from scratch and sort of drag and drop lots of properties in here, but just to save a bit of time, I'm going to take an example profile here, which is looking for appropriate physicochemical and ADME properties for an orally dosed compound against a CNS target, 5-HT1A in this case. So here I'm looking for solubility, good human intestinal absorption, the right range of log P, good blood-brain barrier penetration, avoiding PGP transport, and so on. So you can define these criteria. But of course, what I also want is a potent compound. And so I'm also going to add this potency criteria here, this experimental potency. I can just drag and drop in there. And I can say, oh, maybe I'm interested in a pKi of greater than 7. And of course, that's really, really important. So I'm going to drag the slider up to the top here. So you can very, very easily set up an appropriate scoring profile. Of course, we might want to be more sophisticated. This is a hard cutoff. So I could go in here instead and design a desirability function. So I could actually say maybe between 7 and 8, I'm going to go from my low score to my perfect outcome. And I can define one of these simple trends that I showed you before. So ideally, we want to have better than 8 pKi. We definitely don't want to have worse than 7. And between those, we have a sort of ever-increasing desirability of compounds for that property. And so now you can see I've added that more complex desirability function. I can give this a name. And I can run this against my data set here to calculate those scores. And I could sort my compounds. And now if I sort them into descending order, you see the highest scoring compounds come to the top. And we have some compounds in here that have a 40% chance of success against our criteria here. So that's very good, particularly given the uncertainty when combining all of those predictions and a fairly noisy experimental outcome. If I scroll down, of course, I get to some compounds that aren't looking so good. You can see here, these compounds have only or less than a 10% chance of success. And you can sort of see why as well. If I just expand this out, you can sort of see here that the problem with this is a sort of mint green bar on the left, which is it's very unlikely to achieve our potency criterion. Better than 8 we were looking for, and this is just over 7. On the other hand, if I look at this compound above, compound in row 95, this has great potency. The issue here is the light blue bar, which is the log P. So I don't need to optimize the potency very much here. Really, what I should be focusing on are the physicochemical properties of that compound in order to improve its chance of success. So it provides you that sort of guidance as to what to focus on. Now we can look a little bit more detail at these compounds. I'm just going to jump into what we call card view here. I see what I've done. Excuse me a second. I have mistyped, so it's not picked up the score. Let me just rerun that for a second. There we go. So now we can actually look at each compound individually. We can see the scores for them and the key data. But what I'm actually going to do here is actually to stack these up into the different chemical series that we have here. So what we have here, we just combined all those little cards into stacks. You can see here, each one represents a different chemical series. You can actually see the number of compounds in each of these series here at the top. You can also see a representative structure and the distribution of that potency. So here again, remember the indinol series has lots and lots of really potent compounds. The box plot below shows the distribution of the score, though, and actually, in terms of its overall score, it isn't looking so good. I zoom in a little bit so you can see that maybe a bit better. So there are issues here with those physicochemical and ADME properties, whereas this aminotetraline series has a broader distribution of potency. There's some really potent compounds, some that aren't, but in terms of the overall score, some of those very potent compounds are also very likely to have good physicochemical and ADME properties. Those are those high scoring compounds that we saw. So this might be a really interesting series to look at. But we can look at that in a really rigorous way. Which series should we take forward? Which series should we reject? And so I'm going to go back to the visualization here, and I'm going to create a snake plot like the one I showed you in the slide. So here are the compounds ordered from the best on the left to the worst on the right. Here on the y-axis is that overall score. So there's our best compound with a score of just over 0.4, 40% chance of success. But now again, you can see the error bars, and there's quite a lot of uncertainty here. So really, we can't confidently distinguish between maybe the top 80 compounds in this data set based on the data we have so far. And if I draw around those to select them in the plot, you can see they're selected here, the little blue bars, the buckets fill up around each of these series of compounds. And you can actually see that we should at least consider 4 of these series. There are 2 that we can reject with confidence. They're very unlikely to be as good as the best compounds, but we should at least sample some from each of these series and take them forward to get some experimental data for those ADME properties, which will have greater confidence, so the error bars will become smaller, and then I'll know for sure which ones I should move forward, perhaps into lead optimization. So we can look at this in a very rigorous way in terms of selecting compounds for progression, going from quite a diverse set here into one or a small number of specific series. We can, of course, also use this to guide design if we're trying to improve compounds. So here, if I look at this aryl piperazine series, I've just selected some of these out into a separate data set here where I've already scored them. Here, the only difference is I've included a prediction of the activity in the score, so I can use that to guide design. And if I maybe take this highest scoring compound here, I can go into card view so we can look at that in a little bit more detail. What you can see here is a good score. It's best score for this series. It's a nice, very active compound. But the issue you can see from the pink bar is that the hERG inhibition is higher than we would like. It's better than or worse than 1 micromolar, a PIC50 greater than 6. So I might want to think about, well, can I design new compounds that will reduce that hERG liability, but not sacrifice all of the other properties that I'm looking to achieve for a high quality compound? So I can go to the designer here and I can try some new compounds. I can focus in on improving this overall score. And I could even use the glowing molecule in Stardrop to think about where to change this compound. Here, the red indicates that these regions are tending to increase the hERG inhibition. The blue are tending to decrease it. So I might think, well, basic nitrogen associated with a flexible linker and aliphatic group, so aromatic group, excuse me. I could maybe change the basicity by making that into an amide. I can do that. You can see, yes, I have reduced the hERG inhibition by about 0.6 log units, so that's really good. The potency is still fine. The mint bar here on the left-hand side looks good, but you see the overall score has gone down. So although I fixed or improved one problem, I've created another, which is, you can see the bars here, the orange and the brown, they've gone down. And that is because I'm having a big effect on reducing the blood-brain barrier penetration, which clearly is a critical requirement for a CNS drug. So interesting idea, but that difficult trade-off we often have to face, which is fixing one problem often creates more. So yeah, we can try lots of ideas. We can even start to use generative chemistry to explore this as well. I can say, well, find me some ideas to give me a higher score overall, and maybe I'm willing to replace this end of the molecule. Give me some ideas for replacing that, and away it will go, and it will start to generate suggestions that are likely to improve that overall score of the compound. So balancing all of those factors automatically. So again, a few indications of how, once you can do this rigorous multi-parameter optimization in a convenient way, you can start to apply it to many different questions in terms of the selection and progression of compounds in a project, but also in terms of the design of new compounds. And with that, I will wrap up, leaving just a few minutes for questions at the end. So just in conclusion, hopefully you've seen how multi-parameter optimization enables us to select and design high-quality compounds, quickly targeting compounds with a high chance of success, but also being very careful to avoid missed opportunities. That opportunity cost is often hidden when we throw away potentially good compounds. So to do that, we need to be very conscious of the limitations of the data we have in drug discovery, both in terms of the relevance to the overall clinical outcome, as we saw in that CACO2 example. A poor CACO2 permeability does not necessarily translate into a poor clinical outcome. And of course, taking the uncertainty of that data into account. There's a lot of experimental variability and certainly a lot of statistical uncertainty we need to consider. And so this balanced strategy can dramatically reduce the time and resources required for compound optimization to find those high quality compounds for our projects. And so thank you very much for your time and attention. I see we've already got some questions coming in. I'm going to hand back to Katie, who is going to ask a few of your questions. Again, if we don't have time to get through them all, then we will, of course, reply to you afterwards as soon as possible by email. Over to you, Katie.
Thanks, Matt. Thanks for a well-considered and thorough presentation. Hopefully everyone on the call really enjoyed everything you had to show. So as a reminder, if you do have any questions, please add these to the Q&A panel at the bottom of the screen. And as Matt mentioned, any that we aren't able to answer today, we will follow up with after today's webinar. So the first question is, how do I use MPO if I have missing data?
Yes, so missing data is really important because we don't measure every compound in every experiment, at least not to start with. So yes, we can treat that in a very rigorous way. We talked about dealing with uncertainty in data. Well, a missing value is just a very, very uncertain value. We essentially infinitely uncertain within the range of possible outcomes. And so we can treat that rigorously and look at the effect that has on the overall score and in the confidence in our prediction. So for example, if we had a compound that was perfect for everything, but was missing one value for a very important property, that would show up as a sort of a mediocre score because, we're not really sure, but it will have a big error bar because I measure that value and it's good, it's the perfect compound, so that'll have a very, very high score, a high probability of success. If we measure that very important property and it's a poor outcome, well, we're out of luck. It's not a good compound, and so that's reflected in the size of the error bar. If you look at another example, we might have a compound that actually failed in multiple properties but was missing a value. Well, that missing value is not going to have an effect. It doesn't matter what it is. It's actually going to come up with a low score with high confidence, because measuring that value and getting a good result isn't going to magically rescue the other 4 or 5 parameters it's failed for. And so it does the right thing. It's telling you, don't bother measuring that. Don't waste your time and resources. So it will propagate that uncertainty appropriately.
Thanks, Matt. Another question that's come in is, how important is it to establish the error for every testing area, particularly for biological screening?
Yeah, so I mean, I guess one of the key messages of this presentation is it really is important to take into account the uncertainty in the data, because you could be inappropriately throwing away compounds based on an uncertain outcome. So it is important to use some form of uncertainty, some uncertainty estimate at least. Now, we're not always in the luxurious situation of having multiple replicates and a mean and a standard error in mean for each of our experiments. Equally, we're not always in the fortunate position of having a rigorously determined variability, even in a single point data, but at least a reasonable estimate of the confidence you can have in the values is helpful to use. Even if it's chatting with the biologist who set up and runs the standards on the assay and can at least say, well, what do you consider to be a different outcome if 2 compounds get different values? Would you consider that to be different? And just encoding that as an approximate uncertainty is better than ignoring uncertainty entirely.
Thanks, Matt. Another question, probably the last one for today's session, is what is the scoring model being used to predict change in hERG activity when you added the ketone? Is it an internal data set for training?
Yeah, absolutely. So one of the things that I looked at in this quick example was predicting the hERG inhibition sort of on the fly, and using the glowing molecule here to show the impact of different regions of that molecule on the hERG inhibition. This is a QSAR model that we have developed. It's part of what we call our ADME QSAR module. So it covers a broad range of high-quality models for physical, chemical, and ADME properties. It is based on very carefully curated data, high-quality data, and built with well, we use a variety of different QSAR modeling methods. There's not one method that's always best. I think this one is, off the top of my head, a Gaussian processes model. Yeah, so it's our own in-house model. You can use any model that you like within StarDrop for this MPO. They can be essentially integrated with StarDrop and used alongside our models, or instead of our models for that matter as well. And just to emphasize, you don't have to use predictions. You can use experimental data in this sort of MPO approach.
Thanks, Matt. And actually, there's one final question for an anonymous attendee. So how is this similar or different from QSAR?
Right. So the approach of MPO is, well, first of all, you can apply it to experimental data, whereas a QSAR model is predicting a property based purely on the structure of a compound, quantitative structure-activity relationship. So you can take whatever data you have, experimental or predicted, and combine it against the overall profile of properties that you require. Now, you might simply say, well, I could build a QSAR model that classifies compounds as successful in the clinic or unsuccessful. Well, I'd suggest that's quite a challenging problem to address. First of all, there are many, many different factors that drive success in the clinic. And so your QSAR model would have to be able to correlate the structure of your compound to all of those different factors simultaneously. And unfortunately, we have very few examples of compounds that have a good outcome in the clinic versus many, many examples of things that have failed somewhere in drug discovery or in the clinic. And so you've got a very biased data set. It's very, very hard to build a QSAR model of such a biased data set with so few examples of good outcomes. So this is a way of essentially building a heuristic model that says, given we can measure or predict a set of critical factors for the outcome of our project, can we define that set of criteria that we believe or have statistical evidence are likely to give us that ultimate successful compound downstream? I mentioned one method of doing that, which is the patient rule induction method, but often it is based on experience, slightly subjective, but knowing what you are looking for in terms of these sort of key basic parameters that you're trying to optimize. Another example is this interpretability, whereby, if I have a poor outcome for a score, I can see why. I can see what is the property that's driving that. And often it's much easier to understand the SAR driving that single property to guide the optimization to overcome that outcome. So it's quite different from QSAR in that spec. But as I demonstrated, it can use QSAR models of these individual properties very, very effectively to drive optimization.
Thank you, Matt. That's really helpful. I'm afraid that is all that we have time for today, but if you wanted to learn more about MPO, I'd encourage you to take a look at our website, which has lots of helpful information and links to all of our relevant materials. All that's left for me to do is to thank Matt for a really insightful talk, and thank you all for coming, and we'll see you next time.
Indeed. Thank you all, and thank you, Katie.